2 of our respiratory lecture, where we left off at the trachea, also called the windpipe. And within the trachea, in the mucous membrane, there are cilia. And the cilia, just like we talked about with the nasal passages, um, that they will move mucus with trapped particles towards the pharynx, so does the trachea. And so the goal in the cilia know which direction to move the mucus. They move it towards the pharynx. So this is another way that your body is protecting the tender airways deeper in the lungs from particles of debris and dirt, dust, uh, smoke, um, and well, smoke actually can get past there, but particles that contain bacteria and other kinds of debris. So also the trachea then is composed of C-shaped cartilage rings, and they are there in order to prevent the mucous membrane from collapsing. So the tube is composed of cartilage with then some other connective tissue. And the posterior part of the trachea actually has smooth muscle and fibrous connective tissue in it, which then allows the esophagus to expand when swallowing happens, and it allows it to kind of push into the trachea. So let me get an image to show you. Right here, this is the trachea, and on behind it in this picture would be the esophagus. So if we cut this open and look at the cross section, there's the C-shaped ring of the trachea right here, and there's the mucous membrane here with the pseudostratified ciliated columnar epithelium. And then here's the esophagus, which is a muscular tube that allows the passage of food. We'll learn about it in the chapter 24. But right here, where the trachea would touch the esophagus, there's not cartilage, but there's a muscle, smooth muscle right here, along with the mucous membrane and connective tissue. And this allows so that if the esophagus expands and pushes in, it's not going to be pushing up against hard cartilage. It's going to be pushing up against soft tissue composed of smooth muscle and connective tissue. All right, so we've explained that. Let's go to the next image. This is just showing you again the trachea, and we'll learn about this in the lab, where we have the branching on the right and the left side. These first set of branches are called the primary bronchi that branch off of, pardon me, I don't know why it does that, branch off the trachea. So the trachea, primary bronchi, and then they branch into further branches that are called the secondary bronchi. All right, so in the bronchial tree, which I just mentioned, we have what are called bronchi that have cartilage plates, and they have smooth muscle inside them. The cartilage plates are there to help prevent the tube from collapsing, just like we talked about the trachea is prevented from collapsing because of those C-shaped cartilage rings. So bronchi have cartilage plates with smooth muscle inside them. And then bronchioles are even smaller than bronchi. Those are smaller branches that are not visible. You can only see them microscopically. You cannot see them with the naked eye. And they are composed of smooth muscle in the walls. And the sympathetic nervous system, the stimulation of the sympathetic, will cause relaxation of the smooth muscle in the airways, causing bronchodilation. So this you'll learn, or you should have learned in the sympathetic nervous system, that stimulating certain receptors, in this case it would be the beta 2 receptors in the lungs, which you don't have to know that for this class, for this lecture, but that causes bronchodilation. And parasympathetic stimulation will cause constriction of the smooth muscle in the airways, causing bronchoconstriction. The greatest resistance to airflow in the lungs is due to the bronchioles. Remember the arterioles we talked about, those are also resistance vessels. and that's where the greatest resistance to flow is when they're constricted. There's a lot of resistance, and that helps build up the pressure. Well, in the airways, the bronchioles are where you have the greatest resistance to airflow. So opening the bronchioles a little bit will very much open up your airways and improve the amount of air that's able to pass. And then we have something called terminal bronchioles that will open into alveolar ducts and the alveoli. And here we're getting into the respiratory part of the lungs where respiration actually takes place. Here we go. There's the tail end of the trachea. These would be primary bronchi. This is the left primary bronchus, and these are the cartilage rings here. There's actually some rings very close to the trachea, but then they begin to become cartilage plates, a little bit smaller pieces of cartilage that are there just simply to prevent collapsing of the airway. And through the bronchi, all of the bronchi have cartilage plates that get smaller and smaller. But once you get to the bronchioles, there are no cartilage plates. There's only smooth muscle, and those will ultimately then turn into the terminal bronchiole, the respiratory bronchiole, and then the little alveoli, which are the little air sacs. Here we are showing you just an image of the right lung, the lateral surface, and there are 3 lobes to the right lung. I always like to think of tri-rhi. There's 3 lobes on the right side and 2 on the left side. And we'll learn about those in lab. But let's go ahead and just talk a little bit more about the anatomy of the lungs and how they sit in the body. Each lung is contained in a pleural cavity, and the pleural cavity has a membrane called the pleura that is within it. And just as you remember the pericardium, we had a visceral and a parietal layer, so we have with the pleura. The visceral pleura is a membrane that is on the surface of the lung tissue. It is a serous membrane, meaning a membrane that secretes a watery fluid. And then there's the parietal pleura, which lines the thoracic cavity. That would be your chest cavity. And also it will cover the diaphragm from the inside part of the chest cavity there. It again is a serous membrane that has fibrous connective tissue. There is a cavity between the visceral and the parietal pleura. It's a very small cavity, but there's a little bit of fluid that exists in that cavity called pleural fluid, and that's very important because it decreases the friction between the lungs and the chest wall. And that pleural cavity is kind of interesting in that there's a little bit of negative pressure in that pleural cavity. It's a little bit more low than the pressure that is in the lung cavity. And that little bit of negative pressure is very necessary for ventilation. These pleural membranes actually separate the lungs from each other. So they are in individual tight compartments in the chest. Oops. Okay, this is just showing you a little bit of a lobe of the very small section of lung tissue, a little tiny bit of it, showing you the little air sacs called the alveoli or alveolar, these alveoli, and we'll again take time in lab to go over that. But this is showing you that the visceral pleura lays right over top of the lung tissue, and then there's the parietal pleura, and then the little space in between is the pleural cavity where there's pleural fluid and a little bit of a negative pressure. It's more negative, it's lower pressure than is actually within the lungs themselves. More pictures, images showing you the alveoli, the individual little air sacs that are in the lungs. When you look at tissue, you can see that each of these little passages here is an alveolus. Each of these little sacs that are pointed to here are alveoli, and they will form little ducts called alveolar ducts. and so on. So this is actually a scanning electron micrograph here, and these are just microscopic sections in a light microscope, these two. Okay, this has to be in a, this area is right here is a bronchus that's contained within a lung, and you can see that because there's a little cartilage plate there, so therefore it has to be a bronchus rather than a bronchiole. Bronchioles do not have any cartilage plates, so there's a bronchiole right there. Okay, alveoli. So these are part of really the respiratory portion of the respiratory system. Previously we talked about the conducting portion, which was the nasal cavity and the pharynx and the larynx, the bronchi, bronchioles. But now the alveoli are the functional part of the respiratory system where respiration actually occurs. And the bronchioles will open into alveolar ducts that lead into alveoli and alveolar sacs. So I should actually put into because the word should be there. Now, alveoli have a lining. It's a very thin lining. They have and it's good that it's thin because diffusion has to occur. They're lined with simple squamous epithelium, known as, and the type of cells are called type 1 alveolar cells, and they have a basement membrane underneath them. These alveolar cells are surrounded by blood capillaries, again, which are a single cell endothelial cell lining. This makes for a very short distance for gas diffusion, meaning from the alveolar cell to its basement membrane, to the basement membrane of the endothelial cell, to the endothelial cell. It's a relatively short distance for oxygen and CO2 to diffuse across. I'll show you a picture of that in just a second. Actually, I may do that now if I can find it. That's an image. This is pretty good right here. There's the alveolus in the air space. Here's the alveolar epithelial cell and the basement membrane of, or basal lamina, same thing. The basement membrane of the alveolar cell is fused with the basement membrane of the endothelial cell, which is part of the capillary. So you have this short area for diffusion of gases, and that's, again, a very good thing. It's good that it's a short distance because if it were much thicker, gases would have a problem diffusing across. Endothelial cells are important also in your lung capillaries because they are the source of ACE, or angiotensin converting enzyme. This enzyme catalyzes the conversion of angiotensin 1 to angiotensin 2. And again, this is happening in the alveoli. So, again, that's an essential enzyme in the angiotensin, or the renin-angiotensin aldosterone system. So this enzyme is found in that the endothelial cells. That the endothelial cells produce angiotensin converting enzyme. All right, another type of cell in the alveolus is a type 2 alveolar cell, also called a septal cell, because it's in a septum, or a kind of a wall of the alveoli, in between alveoli. It's called a septal cell, and it secretes surfactant. Surfactant is something we'll be talking about here in just a few minutes. It is a phospholipid that decreases surface tension, and that's very important in breathing, effective breathing. There's a little mnemonic device to help you remember that type 1 epithelial cells here, the type 1 epithelial cells, the simple squamous, have all the fun, meaning diffusion, and type 2 makes the goo, or surfactant. Surfactant is a phospholipid, so it has a, a fun way to talk about it is goo, because it is a phospholipid substance, a little bit sticky. And then the other type of cell in the alveoli is the alveolar macrophage, also called dust cells, and these are phagocytic. They will phagocytize uh, particles and debris, that sort of thing, in the lung tissue. So there's a nice micrograph to show you, a photo micrograph showing you type 1 pneumocytes, which have actually a flatter nucleus here. These are the simple squamous epithelial cells, much more flat nucleus. Probably right here is one as well. And then type 2 pneumocytes have a rounder nucleus here, and then the dust cells are huge, big cells. So they're kind of like sitting right a little bit inside the alveolus. And you can see some capillaries here as well. So there's a question for you. Which cell type would have a lot of smooth ER in the lung? You might want to go back and look at your chapter 3 where it talks about the cell and what the function of the smooth endoplasmic reticulum is. And once you figure out what the smooth endoplasmic reticulum does, then you'll be able to figure out which of these cells would have a lot of smooth ER. Okay, nice images again showing you the anatomy of an alveolus within the lung. So we're getting ready to talk about pulmonary ventilation and the process of breathing. So this is a good time to take a break. Thanks.